deep inside the skull, past the soft folds of the outer brain, past the layered cortex where thoughts form and dissolve and reform in the light of waking, something curves in the dark like a net cast over a smooth and heavy stone. It is not large, it is barely a shell, a thin crescent of tissue pressed close around the thalamus, following the contour of that inner structure, the way a cupped hand follows the surface of something held gently in the dark. The neurons that make up this thin curving layer are small and numerous, and they are always present, and most of what is known about them was discovered not in any single moment of insight, but across decades of slow and careful work in laboratories where slices of brain tissue sat suspended in warm saline under microscope light, while scientists watched what happened when a current passed through. This thin shell is called the thalamic reticular nucleus. The name is old and anatomical and carries with it none of the strangeness of what this structure does. It curves around the thalamus the way a crescent moon curves around nothing, enclosing, attending, never quite completing the circle. And in the deep hours of sleep, when the body has stilled and the eyes have stopped moving and the muscles have let go of the day's accumulated tension, this small curved structure begins to pulse. The pulses are rhythmic. They repeat 11 times a second, or 12 or 13, a range that varies slightly from person to person and from night to night, but always within that narrow sigma band, always locked into that particular frequency, the way a clock is locked to its mechanism. And these pulses travel. They move upward and outward through the sleeping brain, spreading from the thalamus to the cortex through long relay fibers that branch and fan like the veins of a leaf pressed between dark pages. And on the scalp of the sleeper, if a fine electrode is placed at the crown of the head, those pulses arrive as a brief electrical signature, a cluster of waves that rises, crests, and fades in less than 2 seconds, then disappears back into the quiet background of surrounding sleep. These are sleep spindles, and this is where they come from. The neurons of the thalamic reticular nucleus are inhibitory, where most neurons in the brain communicate by exciting their neighbors, passing the signal forward, amplifying, spreading. These neurons do the opposite. Their principal tool is inhibition. The chemical they release into their synapses is GABA, and GABA closes doors. It holds the membrane of the receiving neuron in a state of quiet, below the threshold where firing becomes possible, pressing it gently and firmly downward into a hyperpolarized stillness. To understand why inhibition would produce rhythm, it helps to follow the sequence one step at a time. A thalamic reticular nucleus. Neuron fires. It releases GABA onto a neighboring thalamocortical relay neuron, a different class of cell, one that sits deeper in the thalamic mass, and whose long axon reaches all the way up to the cortex above. The GABA hyperpolarizes that relay neuron, pushing its membrane voltage downward. The relay neuron goes quiet, and for a fraction of a second, a brief window measured in tens of milliseconds, it stays quiet, held there by the inhibitory postsynaptic current that the GABA receptor has opened. But here is where the physics of the membrane becomes important. The thalamocortical relay neurons carry within them a particular class of calcium channel. These channels are called low-threshold T-type channels, and they have an unusual property. When the membrane is held sufficiently below its resting voltage, hyperpolarized, pressed down, these channels become available to open again. They de-inactivate in the darkness of suppression, the way a spring coils tighter as it is compressed. And when the inhibitory current finally fades, when the GABA receptor closes and the hold on the membrane begins to relax, the membrane does not simply drift back to its resting state. Instead, the T-type calcium channels spring open, Calcium flows inward. The membrane voltage rises sharply. The relay neuron rebounds, not into a single spike, but into a brief burst of spikes, a rapid volley of action potentials fired in quick succession before the normal high threshold mechanisms of the neuron regain control and bring the firing to an end. This rebound burst is the engine of the spindle. It is a phenomenon intrinsic to the membrane of the relay neuron, not imposed from outside, not a reflex to any incoming signal, but a consequence of the biophysical properties of the channels that live in that cell's membrane. The depth of the hyperpolarization determines how many T-type channels have de-inactivated. The duration of the hyperpolarization determines the timing of the rebound. And so the inhibitory input from the thalamic reticular nucleus sets not just the fact of the rebound, but its precise timing, the interburst interval that determines the frequency of the oscillation. The relay neuron's burst sends signals in two directions at once, upward, along the long axons that reach the cortex. The burst arrives as an excitatory glutamatergic input, a wave of depolarization that touches cortical neurons and nudges them briefly toward activity, and backward along collateral branches that curve back into the thalamic reticular nucleus. The burst re-excites the very neurons that inhibit it. The thalamic reticular nucleus fires again. The relay neuron is inhibited again. The calcium channels de-inactivate again. The rebound comes again. The loop sustains itself, not indefinitely. The intrinsic properties of the membrane change as the cycle continues. The calcium channels partially inactivate. The GABA receptors desensitize slightly, and after roughly half a second to 2 seconds, the whole oscillation runs down. But for those seconds, it is self-sustaining, a biological clockwork in which each turn of the wheel is caused by the previous turn, in which inhibition produces rebound, and rebound produces inhibition, and the whole assembly spins in the warm, dark interior of the sleeping brain at 11 to 15 cycles every second. This is what the French-Canadian neurophysiologist Mario Steriade spent much of his career studying. Through the 1980s and into the 1990s, his laboratory in Quebec accumulated a body of work on the thalamic reticular nucleus that remains foundational to the field. In slice preparations, thin sections of thalamic tissue kept alive in artificial cerebrospinal fluid, their neurons still capable of firing and receiving inputs. Steriade and colleagues traced the circuit, identified the cell types, mapped the ion channels, and established the TRN as the pacemaker of the spindle rhythm. In intact animals with electrodes placed precisely in the thalamus, they recorded the bursting in real time, watching the inhibition and the rebound play out in the living brain of a sleeping cat. And they observed something that earlier theories had not fully anticipated, that when the thalamic reticular nucleus was experimentally isolated from the rest of the thalamus, the spindle rhythm did not persist. The TRN alone, removed from its reciprocal relationship with the relay neurons, went quiet. The circuit required both partners. This reciprocity is the heart of the mechanism. The thalamic reticular nucleus does not impose rhythm on a passive thalamus, and the thalamocortical relay neurons do not generate rhythm on their own. The rhythm belongs to the space between them, to the loop, to the conversation, to the physical fact of two populations of neurons whose outputs are each other's inputs, bound together by the biophysics of channels that open in hyperpolarization and close in firing. Between the two partners in this reciprocal circuit, the architecture of the thalamus mediates everything. The thalamic reticular nucleus sits at the edge, a thin plate of cells positioned between the cortex above and the deeper thalamic nuclei below. so that virtually every axon traveling between the thalamus and the cortex must pass through it. This anatomical position gives the TRN a kind of passive authority, not as a gatekeeper in any purposeful sense, but because its cells receive collateral inputs from the very fibers that pass through them, and they send their inhibitory outputs back onto the cells those fibers originate from. The geometry alone makes the TRN central to everything that moves between the thalamus and the cortex, which during sleep means everything. The TRN neurons themselves are remarkable in a particular way. They carry both low-threshold calcium channels and high-threshold sodium channels, like most neurons, but the low-threshold channel in TRN cells is not purely the same subtype as in the relay neurons. The biophysics differ slightly, and the result is that TRN neurons can generate their own low-threshold calcium spikes, their own rebound bursts after hyperpolarization, their own intrinsic rhythmicity under the right membrane conditions. The network oscillation and the cellular oscillation reinforce each other. Each cycle of the circuit arrives at a TRN membrane already partially primed for the next burst. Calcium is the messenger that moves the oscillation. In the thalamocortical relay neuron, when the low-threshold T-type channels open and the rebound burst fires, calcium enters through those channels in a brief concentrated current. The calcium current is inward. The ion carries two positive charges, and its entry pushes the membrane voltage sharply positive, triggering the action potential burst. But calcium is also a messenger inside the cell. Once inside, it is sensed by calcium-binding proteins. It activates downstream signaling cascades. It can influence which channels remain available in subsequent cycles. The calcium of the rebound is not simply an electrical event. It is a signal with chemical consequences that extend slightly beyond the immediate millisecond of the burst, shaping the next cycle before it begins. The GABA released by the TRN acts through two types of receptor on the relay neuron's membrane. There are fast receptors, the GABA A type, that open chloride channels and produce a brief hyperpolarizing current lasting tens of milliseconds. And there are slow receptors, the GABA B type, that act through G proteins and potassium channels to produce a longer, more sustained hyperpolarization, one that lasts for hundreds of milliseconds. The GABA B receptor-mediated inhibitory postsynaptic potential is the one that matters most for spindle generation. Its duration, the time the potassium channels remain open and the membrane remains pressed downward, determines how long the T-type calcium channels have to de-inactivate, and therefore how available they will be when the hold finally releases. A longer GABA B potential means a deeper and longer hyperpolarization, means more T-type channels de-inactivated, means a stronger rebound burst, means a stronger return signal to the TRN, means a stronger next cycle. The GABA B receptor is the slow clock within the fast oscillation. None of this is visible to the sleeper. None of it is felt. The neurons cycle through their reciprocal loop in complete silence from the perspective of anything resembling experience, while the body lies in its dark room, the breath moving at its own slow rate, the chest rising and falling without effort, the hands still on the sheets. The only sign that anything is happening, the only trace left at the surface of the skin, is the small electrical field generated by the synchronized activity of millions of neurons all cycling through their inhibition and rebound in close temporal coordination. That field reaches the scalp, and at the scalp, if an electrode is placed at one of the central sites, at the crown, or slightly left or right of center, at the positions a clinical neurologist might call C, C3, or C4, the electrode records a fluctuating voltage that changes shape over the course of the spindle. The first waves are small. The oscillation has just begun, and the network is not yet fully synchronized. The contributing neurons are slightly out of phase with each other, and their individual contributions partly cancel. As the oscillation continues and the loop runs through its second, third, fourth cycle, the synchrony grows. More neurons are entrained. Their contributions add rather than cancel. The amplitude of the scalp voltage grows. This is the waxing phase. On the tracing of the electroencephalogram, the continuous record of the brain's electrical activity printed to paper or displayed on a clinical screen, the spindle appears as a cluster of waves that grows visibly in height over the first half second or so. The waves are sharp and regular, their frequency visible even to the eye as a tight repeating oscillation, nothing like the slow rounded waves of deep sleep or the irregular low amplitude flutter of the waking trace. The spindle looks distinct. Even a student learning to read EEG for the first time can usually recognize one. There is the flat, quiet background of surrounding non-REM sleep, and then, rising from that background, the spindle. The peak is brief, the oscillation crests, the waves are tallest, the synchrony is maximal, the TRN-TC loop is running at its most coherent, and then the balance begins to tip. The calcium channels in the relay neurons begin to partially inactivate, not recovering as fully between cycles as they did at the start. The GABA receptors at the relay neuron membrane begin to show the mild desensitization that comes from repeated activation. The rebound bursts grow fractionally weaker. The return signal to the TRN is fractionally smaller. The next inhibitory volley is fractionally shorter, and the synchrony that built across the first half second begins to dissolve. This is the waning phase. The waves on the EEG trace grow shorter. Their amplitude decreases over the second half of the spindle, the waveform tapering gradually rather than cutting off sharply, a slow release rather than a termination. The oscillation does not stop. It diminishes. The neurons cycle on, but their contributions cancel again as synchrony fades, and the recorded signal at the scalp grows quieter and quieter until it drops below the noise floor of the background and disappears. What is left on the trace is a characteristic shape, a cluster of 11 to 16 oscillations per second, perhaps 10 or 12 cycles in total, rising from background, cresting and fading back. The whole event lasts somewhere between half a second and 3 seconds, and a typical spindle from a healthy young adult in the middle of a night of good sleep might last roughly a second and a half, long enough to be unmistakable, brief enough that it fits entirely within a single breath. The automated detection algorithms that modern sleep laboratories use to find spindles in overnight EEG recordings look for exactly this shape, translated into numerical operations. The raw signal is filtered to isolate the sigma band, the 11 to 16 Hz range, and then the amplitude envelope of the filtered signal is calculated, a measure of how much power is present in that frequency range moment by moment through the night. The threshold for declaring a spindle event is typically set at 1.5 to 2 standard deviations above the mean of the envelope across the full recording. When the envelope crosses that threshold and stays above it for at least half a second, but no more than 3 seconds, the algorithm marks the event as a spindle and records its onset time, duration, peak frequency, and peak amplitude. Over a full night of 8 hours, a healthy young adult might accumulate several hundred of these events, clustered densely in certain stages of sleep, and sparse or absent in others. But the algorithm is working backward. In a sense, it finds the trace that the circuit has left. The circuit itself, the TRN neurons, and the thalamocortical relay neurons cycling through their reciprocal loop in the dark interior of the thalamus, the calcium rushing inward with each rebound, the GABA pressing each relay neuron into its brief silence before the spring releases. The circuit is indifferent to being measured. It does not produce its oscillation for the electrode. The electrode simply happens to be close enough, and the synchronized activity of enough neurons happens to be coherent enough that the field reaches the scalp and makes itself readable. The thalamus itself is not visible from the outside. It sits at the center of the brain, deep below the cortex, above the brainstem, surrounded on all sides by other structures. In a brain removed from the skull and held in both hands, it is the size of a large olive, or two olives pressed together, and its surface is smooth and gray and gives no hint of the complexity of its interior. The thalamic reticular nucleus on the surface of that structure, the thin crescent of inhibitory neurons that generates the spindle rhythm, is so thin that it is difficult to see with the naked eye in post-mortem tissue. It takes the magnification of a microscope and the specific staining of certain cell types to make it visible as a distinct layer. Under that magnification, the GABAergic neurons of the TRN look like most cortical neurons, roughly spherical cell bodies, 2 to 4 major dendrites branching outward, an axon reaching inward toward the thalamocortical relay neurons below. They are not larger than most neurons. They are not more complex in their individual morphology. What makes them functionally singular is their position, that thin plate between the internal capsule and the lateral surface of the thalamus, precisely where every thalamocortical axon and every corticothalamic return axon must pass, and their intrinsic biophysical properties. The low threshold calcium channels that make rebound bursting possible, the GABAergic output that enforces inhibition, the specific connectivity that makes them simultaneously the source of spindles and the recipient of the signals that sustain those spindles. A single TRN neuron, activated in isolation, would not produce a spindle. It would fire, inhibit its target relay neuron, receive a rebound signal back, and perhaps fire again. But without the recruitment of neighboring TRN neurons, without the broad synchrony that comes from many cells entering the inhibitory rebound cycle together, no coherent field would form, no scalp signal would emerge, no spindle would appear on the EAG. The spindle is a population phenomenon. It requires the simultaneous engagement of many TRN neurons, firing in close temporal coordination, inhibiting many relay neurons simultaneously, generating rebound bursts that return to many TRN neurons at once and continue the loop at a population level. How that population synchrony is established and maintained is part of what made the early work on isolated thalamic slices so illuminating. In those preparations, in which the intact anatomical network of the thalamus could be preserved in a thin slice of tissue, kept alive in a dish of warm ionic solution, Sterriade and colleagues observed that spontaneous spindle-like oscillations arose without any external stimulation. The circuit, isolated from the cortex above and the brainstem below, from all the signals that would normally modulate it during waking, could generate its own rhythmic spindle-like activity in the silence of the preparation dish. This showed that the spindle was not imposed on the thalamus from above or below. The rhythm was intrinsic. It was the thalamus's own natural mode of oscillation when freed from the modulatory influences that keep the thalamocortical system in its waking state. During waking, the brainstem sends upward to the thalamus a constant supply of neuromodulatory signals, acetylcholine, norepinephrine, histamine, that maintain the thalamocortical relay neurons in a slightly depolarized state, above the range in which the low threshold T-type calcium channels are available. When the membrane rests above a certain voltage, even a brief hyperpolarization may not push it deep enough or long enough for the calcium channels to de-inactivate fully, and the rebound is weak or absent. The spindle rhythm cannot sustain itself. The circuit stays in its relay mode, faithfully transmitting sensory information upward to the cortex as the eyes and ears and skin report on the outside world. In sleep, these brainstem modulatory inputs fall quiet. Acetylcholine levels in the thalamus drop. The membrane of the relay neuron rests at a slightly more hyperpolarized level. The T-type calcium channels, held near their resting voltage, sit in a region of partial availability. When the TRN fires and inhibits the relay neuron, pushing it deeper still, the channels de-inactivate more fully. The rebound, when it comes, is stronger. The loop becomes self-sustaining. The spindles begin. This transition from the waking relay mode to the sleeping oscillatory mode is not a single moment. It drifts as sleep deepens and the neuromodulatory inputs grow quieter. The thalamocortical circuit moves gradually from one regime into the other, like a pendulum slowing from large irregular arcs into a steady controlled swing. The first spindles of the night are often brief and somewhat irregular. As the depth of non-REM sleep consolidates, the spindles become more frequent, more regular, more characteristic in their waxing and waning shape. And through all of this, the cortex participates. The long relay axons that carry each spindle burst upward from the thalamus, do not arrive at a passive recipient. The cortex has its own intrinsic oscillatory properties, its own bistable tendencies, the slow alternation between a period of depolarization and collective firing, and a period of hyperpolarization and near silence that characterizes deep non-REM sleep. When a spindle burst arrives from the thalamus during the depolarized upstate of a cortical neuron, the excitatory input is amplified. The cortical neuron fires, and the cortical firing sends its own return signal back down to the thalamus along cortical thalamic axons, not just to the relay nuclei, but also via collateral branches back to the TRN, further engaging the inhibitory rebound loop. The cortex, then, is not simply a receiver, it is a participant, sending its own activity back into the thalamic circuit and shaping the dynamics of what comes next. The spindle is a thalamic rhythm that extends itself into the cortex and receives the cortex's reply. The waxing and waning waveform on the EEG is the record of this exchange, not just of thalamic output arriving at the scalp, but of the full thalamocortical circuit cycling through its oscillation and drawing the cortex into the rhythm. At the moment the spindle crests and begins its descent, the amplitude waning, now on the EEG trace, the synchronized bursting in the relay neurons growing weaker with each cycle, the rebound smaller, the TRN firing less forcefully, the synchrony dissolving, the cortex receives one last attenuated volley from the thalamus and sends a last return signal downward, and then the loop runs down completely. The relay neurons rest at their membrane potential, no longer being driven into rebound. The TRN neurons idle between their sparse background firing. The scalp electrode records the flat, quiet background of surrounding non-REM sleep. The waxing and waning shape is complete. 12 oscillations, perhaps 15, rise and crest and fall, the whole event contained within the time it takes to exhale a long, slow breath. On the EEG screen, if one were watching, the spindle would appear and disappear almost before the eye could follow it, a brief tight cluster of waves surfacing from the low flat background, its internal frequency visible as a precise and regular repeating, its beginning and end both gradual, its shape symmetrical in the way of a thing that assembles and disassembles by the same mechanism, running forward, and then slowly in reverse, and then the background again. The slow, low undulations of surrounding deep sleep, the quiet trace that holds no fast rhythm, no tight repeating oscillation, only the long and gentle waves of a brain that has withdrawn from the world and settled into the oldest and most fundamental mode of rest. The thalamic reticular nucleus, that thin crescent of neurons curved around the thalamus in the sealed dark interior of the skull, continues to pulse. Not continuously, the next spindle will not begin for several seconds, perhaps longer, and between this spindle and the next, there is a quiet interval in which the circuit idles and the EEG stays flat. But the conditions that produce the first spindle are still in place, the brainstem modulators still quiet, the relay neuron membranes still resting in their hyperpolarized range, the T-type calcium channels still available, the loop between the TRN and the thalamocortical relay neurons still intact. When the next spindle begins, it will begin the same way the last one did. A TRN neuron will fire. GABA will cross a synapse. A relay neuron will be pressed into silence. The T-type channels will deactivate in the dark of hyperpolarization, and the rebound will come. A brief, precise burst of calcium-driven action potentials, a signal sent upward to the cortex and backward to the TRN, and the loop will turn again, and the waves on the EEG will begin to rise. Small, coordinated, invisible to everything but the most sensitive instruments, repeated hundreds of times across a single night of ordinary sleep, in every healthy sleeping brain, in every bedroom and ward and dormitory room, where the body has found its rest and the outer world has gone quiet. The thin crescent of neurons curves around the thalamus and sends its rhythmic pulses upward through the brain, and the EEG trace captures the electrical shadow of those pulses as a waxing cluster of sigma band waves, and the waves rise for half a second, crest and fade, and the trace returns to silence, and the oscillation is complete. Quiet, precise, self-terminating, always in the same gradual way, not a cut but a release, the amplitude descending by its own diminishing mechanism back to the flat still line that surrounds it, the waves growing smaller one by one until the last one is indistinguishable from background and the spindle has ended, and the sleeping brain rests again in the deep, unhurried dark. A night of sleep, laid out in its entirety, looks something like a range of low hills seen from a great distance. The first hills are the tallest. They rise steeply in the early hours and descend again into brief shallow valleys before the next rise comes. Moving toward dawn, the hills grow lower and the valleys between them flatten out, and the whole landscape settles gradually into something quieter and less dramatic, though never completely still. 4 hills, sometimes 5, sometimes 6, each one taking roughly an hour and a half to complete. The body travels this landscape every night in the dark without knowing it is traveling at all. This is the ultradian rhythm of sleep, the deep structural pattern beneath everything else the sleeping brain does. Ultradian means shorter than a day. It does not mean brief. Each cycle is long enough to hold a great deal inside it, and the architecture within each hill and valley is more intricate than the outline suggests from far away. Move closer, and the smooth hills resolve into layers. At the base of each early hill, pressed deep into the night, there is slow wave sleep, the slowest, deepest, most restorative of the night's territories. Above it, lighter and more varied, lies a broad middle region through which the brain moves more quickly. And at the crest of each hill, Before the brief REM valley, the brain passes through a particular kind of shallow sleep that will occupy much of what follows here. Approximately 4 to 6 times each night, healthy adults complete one of these cycles. The whole night, in a young adult sleeping 8 hours, divides roughly into this repeating pattern. In the first cycle, the descent into slow-wave sleep is steep and prolonged. The second cycle brings another slow-wave period, slightly shorter. By the third and fourth cycles, slow-wave sleep has mostly receded. The hills are lower, the deep valleys gone, and the shallower sleep of the middle region stretches further, occupying more of each cycle's duration. Meanwhile, the brief REM valleys grow longer. The whole landscape slowly tilts across the night, trading depth for breadth, slow-wave richness for lighter, more varied activity. Within this broad architecture, two landmarks matter above all others for what the sleeping brain is quietly doing in the dark. One is stage 3, the deep slow-wave territory of the early night. The other is stage 2, the lighter region that surrounds and bridges the deeper material, the terrain the brain crosses on its way into and out of every slow-wave episode, and the terrain that persists across all cycles, even the later ones, when slow-wave sleep has all but disappeared. Stage 2 constitutes roughly half of total sleep time in a healthy young adult. It is not a transition zone, not a waiting room. It is the most abundant sleep stage in the entire night, and it carries within it the events this entire narration is built around. Sleep spindles occur most densely during stage 2. This is their primary home, their highest concentration, their characteristic territory. A healthy young adult generates somewhere between 4 and 8 spindle events per minute during stage 2. Brief rhythmic bursts, each lasting half a second to 3 seconds, each one a tight cluster of electrical waves, cycling between 11 and 16 times per second. They appear on an EEG recording as small waxing and waning formations rising above the low flat background of surrounding sleep, cresting, then tapering back. In a full night, across all the stage 2 periods combined, hundreds of these events accumulate. They are not exotic, they are not rare. They are among the most frequent patterned events in the sleeping brain, distributed across the night in a way that follows the architecture of the cycles themselves, plentiful in the first cycle, present through all the rest, changing in subtle ways as the night progresses. To understand why that distribution matters, it helps to stand for a moment inside the landscape of a single cycle and observe how the spindle-generating conditions rise and fall. Each cycle begins with the descent from wakefulness or light sleep into the slow-wave territory. As the brain deepens into stage 3, something else is happening at a larger scale. Large, low, rolling waves are moving through the cortex at less than 1 cycle per second, sweeping across millions of neurons in rhythmic coordinated surges. These are the cortical slow oscillations, the broadest electrical rhythm of deep non-REM sleep. They are not subtle. On an EEG record, they appear as enormous landscape features, wide, gentle undulations that dwarf the faster activity nested within them. In the up phase of each slow oscillation, when the cortex briefly surges into collective activity, the conditions for spindle generation are at their most favorable. During stage 3, spindles do not disappear. They nest inside these slow waves, riding the up phase, fitting themselves into the temporal window of cortical excitability that the slow oscillation opens and closes with each cycle. The image of a spindle riding the up phase of a slow oscillation is not a metaphor. On a high-density EEG recording, the kind that places dozens of electrodes across the entire scalp, The two rhythms are visibly coupled. The slow oscillation makes the first large movement, its rising phase lifting the EEG trace upward. Then, on that ascending slope, a faster cluster of waves appears, tight and rhythmic, the spindle burst arriving precisely within the window of up-phase excitability, before the slow oscillation descends again into its down-phase silence. The spindle fits inside the slow wave, the way a brief tremor sits within a larger ground movement. The slow wave does not cause the spindle in the sense of mechanically producing it. The spindle generating circuit has its own internal machinery, its own reciprocal oscillator operating at thalamic and cortical levels. But the slow oscillation gates the window during which that oscillator is permitted to fire. When the slow oscillation is down, the cortex is in a state of collective hyperpolarization and spindle activity is suppressed. When the slow oscillation rises, the gates open and the spindle circuit can engage. This timing relationship was established through careful human research, particularly through work using high-density EEG in sleeping participants, which allowed researchers to track the spatial and temporal relationships between the two rhythms simultaneously across the entire scalp surface. What emerged from that work was not a loose statistical tendency, but a precise phase preference. Spindles clustered on the rising slope and near the peak of the slow oscillation's up phase, not randomly scattered across the oscillatory cycle. The brain is not indiscriminate about when it generates these bursts. Within the architecture of slow-wave sleep, spindle activity is organized in time, moving out of stage 3 and back toward stage 2. The slow oscillations fade, their amplitude decreases. Their frequency, already well below 1 per second, becomes less regular. The broad rolling landscape of deep slow-wave sleep gives way to the more varied terrain of lighter non-REM. And here, in stage 2, spindles no longer need to nest inside slow oscillations to occur. They arise more freely, more continuously, more densely per minute than during stage 3. The nesting relationship is loosened, not severed. Slow oscillation-like activity persists at reduced amplitude even in stage 2, and spindles still show a statistical preference for upstate-like periods, but the dominant pattern shifts. In stage 2, spindles are the prominent feature. The background is their backdrop, not their container. There is another waveform in stage 2 that intersects with spindle activity in a precise and important way. A K-complex, one of the defining features by which stage 2 is identified, appears on an EEG trace as a distinctive two-part structure, unlike anything else in the sleep record. First comes a sharp, steep negative deflection, a sudden downward spike in the electrical potential. It is brief and pronounced, the kind of waveform that catches the eye of a scorer working through hours of otherwise undulating background activity. Then, immediately following, comes the second component, a broad, slower positive curve upward, a wide wave, completing itself over roughly half a second. The whole complex is large and asymmetric. The spike downward is faster and sharper than the slow curve that follows it. Together, the spike and the curve resemble something like a compressed version of the slow oscillation, a down state followed by an up state, occurring in faster and more concentrated form. What happens at the trailing edge of a K-complex as its positive curve begins to rise is not accidental. Spindles cluster there. The upward slope of the K-complex's positive component creates a period of cortical depolarization, a brief up state, and the spindle-generating circuit responds to that up state in precisely the same way it responds to the up phase of a slow oscillation in deeper sleep. A spindle begins, It waxes through its first few cycles, crests somewhere near the peak of the K complex's positive component, and then tapers as the K complex resolves. On an EEG trace, the two structures fit together at their boundary like adjacent pieces of a biological mechanism, the K complex's sharp spike and broad curve giving way, smoothly and immediately, to the tight rhythmic oscillations of the spindle. The transition between them is so consistent that detecting one often predicts the imminent arrival of the other. Spindles that occur on the positive slope of K complexes are not identical in their properties to spindles that arise spontaneously between K complexes. Research examining the functional significance of spindle timing relative to K complexes suggests that these K complex associated spindles carry particular relevance for the cortical processing that occurs during NREM sleep. The K complex itself appears to reflect a brief, spontaneous cortical upstate that the sleeping brain generates in response to sensory input, a sound, a movement, a thermal change, as well as internally without any external trigger. It represents a brief period of organized cortical activity within the otherwise quiet background of stage 2, and a spindle arising on its ascending slope arrives precisely when the cortex is most receptive, most briefly active, most prepared to engage in the rapid synchronized exchange that spindle-generating circuits make possible. The distinction between fast and slow spindles, a distinction that will matter considerably here, maps partially onto geography across the scalp. Fast spindles, cycling closer to the upper end of the sigma band, around 13 to 16 cycles per second, tend to predominate over central and parietal electrode sites, particularly over the regions of the scalp that overlie sensory motor and parietal association cortex. Slow spindles, cycling in the lower part of the sigma band, around 11 to 13 cycles per second, tend to predominate more frontally over the prefrontal regions. The difference is not absolute. Both types occur at all sites to varying degrees, but the geographic tendency is consistent enough to be replicated across many studies and to form the basis for meaningful distinctions in functional interpretation. What the brain is doing with a fast parietal spindle is not identical to what it is doing with a slow frontal spindle, and the spatial distribution of spindle activity across the night maps onto the geography of the memory processes being served. Across the night's ultradian cycles, the distribution of spindle activity is not uniform, but it is also not simply declining or increasing. It shifts. In the first two cycles, slow wave sleep is prominent, and spindle nesting within slow oscillations dominates the deep periods. As the early night slow wave activity recedes across the third and fourth cycles, stage 2 stretches out between the increasingly brief slow wave episodes and the increasingly long REM periods. Spindle density within stage 2, events per minute, does not simply fall off as the night progresses. Some measures of spindle activity show sustained or even slightly elevated density in the stage 2 periods of the middle and later cycles, when slow wave sleep has retreated and stage 2 occupies a greater proportion of each cycle. The night spindle production, totaled across all cycles, accumulates in a pattern that follows the changing dominance of stage 2 itself, plentiful early, persistent throughout, somewhat concentrated in the long stage 2 stretches that fill the second half of the night. This matters because the consolidation processes that spindle activity supports are not instantaneous. They unfold over time. They require repeated cycles of spindle activity to accumulate. A single spindle burst, however precisely timed, does not by itself produce a durable memory trace. What the night provides is not a single event, but hundreds of events, each contributing its small increment to the molecular and synaptic changes that transform a fragile new memory into something stable. The architecture of the night, the repeating cycles, the multiple stage 2 episodes, the sustained availability of the spindle generating circuit across all 6 cycles creates the temporal scaffolding within which that accumulation can occur. Imagine a single NREM episode from the middle of the night, roughly 3 hours after sleep onset. Slow wave sleep has been briefer in this cycle than in the first. Stage 2 has already occupied most of the NREM portion, the spindle-dense territory stretching out on either side of a short slow wave period. Within this Stage 2 stretch, spindles are not evenly distributed. They cluster. 5, 6, 7 spindle events appear within a span of a minute or two, each one arising and resolving over its half-second to 2-second duration, each separated from the next by only a few seconds of relative quiet, then a longer quiet interval, then another cluster, then quiet again, before the brain edges toward REM period. These clusters are not random. Within NREM episodes, spindle events organize into brief high-density periods separated by relative quiescence, a pattern that is more pronounced early in the night, when slow-wave activity is richest, and the nesting of spindles within slow oscillations creates natural concentrations. But even in the later, lighter NREM episodes, the clustering tendency persists. The spindle-generating circuit does not fire at a perfectly uniform rate across the entire NREM episode. It fires in concentrated bursts of activity, windows of heightened oscillatory engagement lasting minutes, surrounded by quieter intervals of lower spindle probability. What this creates is not a continuous background of spindle activity, but a series of concentrated windows, each one a brief period during which the thalamocortical circuit is most actively cycling through its rhythmic exchange, most densely generating the electrical events that will accumulate across the night into something the morning brain can use. The waveform of the K-complex deserves one more moment of attention before moving on, because its role in organizing Stage 2 activity is more active than passive. The American Academy of Sleep Medicine defines Stage 2 NREM by the presence of spindles and K-complexes in the EEG record, not one or the other, but both as characteristic features of this territory. The K-complex has been studied since Hans Berger's early EEG recordings in the 1930s, though its functional significance has taken decades longer to clarify. For a long time, it was treated primarily as an artifact of the scoring system, a useful landmark for identifying Stage 2, but not itself a mechanism of particular interest. What emerged through more careful study is that K-complexes are not passive background features. They are spontaneous cortical responses generated internally or triggered by low-level sensory events that reach the cortex without waking it, and they represent the same bistable cortical mechanism that generates slow oscillations in deeper sleep, operating at a reduced amplitude in the lighter NREM territory of Stage 2. The down spike of the K-complex corresponds to the down state of the slow oscillation, the moment of collective cortical hyperpolarization, the quiet phase, the period when most cortical neurons are briefly held in silence. The upward positive wave corresponds to the up state, the brief collective depolarization, the active phase, the moment of organized cortical engagement. Seen this way, a K-complex is a truncated slow oscillation, one cycle of the B-stable rhythm, appearing in isolation within Stage 2, rather than in the long, slow, repetitive series that characterize Stage 3. And the spindle that arrives on its ascending positive wave is doing what Stage 3 spindles do at a larger scale. engaging the thalamocortical circuit precisely during the cortical upstate, precisely when the conditions for synaptic engagement are most favorable. The architecture of the night, then, is not just a matter of how long each stage lasts or how many cycles occur. It is a matter of how the different rhythms nest inside each other, how the slow oscillation gates the spindle in deep sleep, how the K complex creates isolated upstates in lighter sleep, how spindle generating activity concentrates into brief clusters across each NREM episode. The full night is a layered temporal structure, and spindle activity is distributed within it according to the opening and closing of conditions that favor thalamocortical oscillation. When the slow oscillation rises, the spindle has a window. When the K complex creates an upstate, the spindle has a window. When the stage 2 period stretches out across the middle and later cycles, that territory is not emptied of spindle generating capacity. It is, in a sense, where the sustained accumulation of spindle events across the night is most consistently maintained. The shift in the night's composition from its first half to its second half is visible in the EEG record as a slow change in the dominant rhythms. In the early hours, the broadband low-frequency power is high. The slow oscillations and delta waves of deep, slow-wave sleep fill the spectrum. Moving toward dawn, that low-frequency power gradually decreases. The sigma band, the 11 to 16 cycles per second range that spindles occupy, does not disappear as the night progresses. In some participants, sigma power actually shows a slight increase in relative prominence during the second half of the night, when deep slow-wave activity has receded and stage 2 has become the dominant NREM territory. The total spindle count accumulated across the full night reflects this distribution. Substantial early, when nesting within slow oscillations creates organized clusters. Persistent later, when stage 2's extended duration keeps the spindle generating circuit active across the long, middle, and final cycles. A single night, then, provides not 1 but 4 to 6 opportunities for spindle-rich NREM episodes. Each opportunity is shaped by its position in the cycle, by the depth of the slow-wave sleep that precedes or follows it, by the density and amplitude of slow oscillations or K complexes that gate the spindle-generating windows within it. The total spindle budget of the night is distributed across these opportunities in a way that no single cycle fully provides on its own. This is in part why truncating sleep, losing the later cycles, the ones that carry the long stage 2 stretches, and the extended REM periods, removes not merely a small fraction of total spindle production, but an entire category of spindle activity distributed across the final third of the night. It is also worth noticing that the spindles of the later night are not identical in character to those of the earlier night. Research examining spindle properties across the ultradian cycle has found subtle but consistent shifts in spindle frequency, amplitude, and spatial topography as the night progresses. Early night spindles, generated within and around slow-wave-rich episodes, tend to show certain features, particularly in their coupling to slow oscillation phase, that differ from the spindles of the later stage 2 dominated cycles. The circuit is the same, the waxing and waning form is the same, but the context in which each spindle fires, the oscillatory environment surrounding it, shifts across the night in a way that is not merely a matter of quantity. The night is not simply a fixed budget being spent down. It is a changing landscape through which the spindle-generating circuit continues to operate, adapting to the conditions of each successive cycle. Deep in the final third of the night, roughly 6 hours after sleep onset, a stage 2 NREM period stretches out between 2 long REM episodes. The slow-wave valleys are gone from this part of the landscape. There may be occasional brief slow wave-like activity, but the deep stage 3 of the early night has not returned. The EEG record at this point shows the characteristic stage 2 mix, a low amplitude mixed frequency background interrupted by K-complexes and spindles. A K-complex appears, the sharp downward spike, then the broad upward curve, and on the ascending positive slope of that curve, a spindle begins. 11, 12, 13 cycles per second, the waveform waxing over the first second, holding briefly at its crest, then tapering back into the quiet background. The event lasts less than 2 seconds, then quiet for several seconds, then another K-complex, another upward slope, and the same brief rhythmic burst arising and resolving in the dark. These late-night spindles are among the most numerous the brain generates in a full night of sleep, simply because stage 2 is so prevalent in the final cycles. Each one is generated by the same thalamocortical circuit, the same thin shell of inhibitory neurons casing the thalamus, the same reciprocal exchange between thalamus and cortex that has been cycling through this rhythm since the first NREM episode of the night. The circuit does not tire. It does not slow. It continues cycling through its rhythm in each successive NREM episode, generating the same waxing and waning bursts that mark stage 2 sleep from its beginning to its end. In each NREM episode across the night, the spindle events accumulate in clusters separated by quiet intervals. The clusters are brief, a minute or two of elevated spindle density, then several minutes of lower activity, then another cluster. Within a single NREM episode of perhaps 40 minutes, 3 or 4 such clusters might occur, each one a brief concentrated period of oscillatory engagement that then subsides. The quiet intervals between clusters are not inactive. Other processes are occurring. The cortex is not still. Slow oscillations or their remnants continue to shape the electrical landscape. But the spindle-generating circuit has its own rhythm within the episode, its own pattern of engagement and relative quiescence, its own clustering tendency that creates brief concentrated windows rather than a uniform continuous background of sigma band activity. Across the full night, these clusters accumulate. In Renwarko, taken together, the spindle events from all NREM episodes, from all cycles, from the K-complex associated bursts, and the slow oscillation nested bursts, and the freely occurring stage 2 bursts of the late night, constitute a sustained and repeated pattern of thalamocortical oscillation distributed across hours. The pattern is not random. The distribution is not accidental. It follows the architecture of the ultradian rhythm, the nesting of spindles within slow oscillations, the gate opened by each K-complex's ascending slope, the concentration of stage 2 sleep in the cycles that bridge the slow wave-rich early night with the RM-rich final hours. The landscape of the night, viewed from the perspective of spindle activity, is a precisely organized temporal structure, not a smooth background hum, but a series of concentrated windows, opening and closing across the hours, each one brief, each one adding its increment to the accumulating work of the night. The last cluster of spindles in the final NREM episode of the night occurs perhaps an hour before waking. The stage 2 period is thinning. The final NREM episode is approaching. A few last K-complexes appear, each one followed by its characteristic brief spindle. The waveforms rise and crest and taper in the same form they have taken all night, the same 11 to 16 cycles per second, the same waxing and waning envelope, the same half-second to 2-second duration. Nothing in their appearance distinguishes them from the first spindles of the night. The circuit has been running this rhythm for 7 hours. These last few bursts arrive and resolve like all the others, quietly, completely, leaving the EEG trace to settle back into the low, flat background of surrounding NREM sleep as the final cycle approaches its end. The architecture of the night delivers sleep spindles not as a single event, not as a continuous background, but as a series of brief, concentrated clusters distributed across every NREM episode of every cycle, organized by the nesting of faster rhythms within slower ones, shaped by the opening and closing of cortical windows that slow oscillations and K-complexes provide, and sustained across the full duration of the night by the persistence of stage 2 sleep through all 4 to 6 cycles from the first hour to the last. The clusters are short. Each one lasts only a minute or two before subsiding, but repeated across the night, across the dozens of NREM episodes, the hundreds of individual spindle events, they form a sustained and precisely patterned contribution to the sleeping brain's nightly work. Each cluster a brief window, each window a concentrated period of oscillatory engagement, each engagement adding its quiet increment to the slow accumulation of the night. 3 rhythms move through the sleeping brain at once, nested inside each other like rings of water inside rings of water, each one turning at a different speed, each one fitting exactly within the time the one above it allows. The slowest is barely a rhythm at all. It moves like a tide, completing less than 1 full cycle every second, its waves so broad and unhurried that it takes almost a full second just to rise from its lowest point to its highest. This is the cortical slow oscillation, a rolling electrical shift that sweeps across the surface of the sleeping brain in a slow front, carrying millions of neurons from silence into brief coordinated firing and back into silence again. It does not announce itself. It has no sound the sleeping person could hear, but in the darkness behind the eyes and beneath the skull, in the thick gray folds where waking thought lives, it rolls through like a deep swell moving underwater that is otherwise still. Inside that swell, riding on its rising slope, a faster rhythm stirs. 11 times each second, or 12, or 14, the thalamocortical circuit sends a brief synchronized burst to the cortex, and for half a second, or a little more, this faster tremor sits atop the slow wave, the way a smaller wave sits on the shoulder of a larger one at sea. This is the sleep spindle. It is brief. It lasts perhaps 1 second, rarely as long as 2, and then it is over, and the slow oscillation continues its long arc, carrying no visible trace of the spindle that just rode across its back. Inside the spindle, something faster still. Down in the curved structure of the hippocampus, in a region called CA1, a ripple fires. It lasts 50 to 100 milliseconds, a tenth of a second, less sometimes, and in that window it oscillates at 80 to 100 cycles each second. If the spindle is a tremor on a wave, the ripple is a shiver inside the tremor. It is so brief, so contained within the moment the spindle has opened, that its entire life would be invisible to the naked eye on an unscaled recording. Only instruments calibrated to detect changes in voltage at the scale of millionths of a second can show what is happening, and what is happening in that fractional window is a replay. The replay is the reason the 3 rhythms are nested in the precise order they are. It is not chance that the ripple falls inside the spindle, and the spindle falls on the up phase of the slow oscillation. The timing of each rhythm creates a window for the one inside it, a narrow opening of the right electrical conditions, the right membrane states, the right balance of excitation and inhibition, so that the fastest and most information-dense of the 3 can fire at exactly the moment its contents can be received. A hippocampal sharp wave ripple carries compressed sequential replays of prior waking experience. This was first established in rodents, in experiments where electrodes implanted in the hippocampi of sleeping rats could record the firing of individual cells with enough precision to identify which cells were active, in what order, and for how long. During waking, when a rat moved through a maze, certain neurons fired in sequence, one after another, as the animal moved from turn to turn, each cell corresponding to a position in space. These cells were place cells, and the pattern of their firing over several seconds built a kind of internal map of the path taken. Then the rat slept, and in the subsequent slow-wave sleep, in the quiet intervals between waking, the same sequence of cells fired again, not over several seconds as it had during the maze, over 80 milliseconds. The entire experience of the path, the ordered chain of spatial positions, compressed into less time than it takes to blink, and replayed without the animal moving a muscle. This compression is the sharp wave ripple. The sharp wave, a large slow depolarization generated in the CA3 region of the hippocampus, arrives at CA1 like a push from behind, and the cells in CA1 respond by firing in the rapid synchronized burst that constitutes the ripple itself. Inside that burst, in the order dictated by their prior waking activation, the cells that encoded the experience fire again. The sequence is intact. The temporal relationships are preserved. The content of the experience is there, packed into a fragment of a second, played back to nobody and to everybody, to the synapses and circuits that are listening, which is to say, to the cortex that is ready to receive it. The cortex is ready because the spindle has prepared it. In the roughly 50 to 150 milliseconds before a hippocampal ripple fires, the corresponding region of cortex passes through a spindle trough, the falling phase of the spindle cycle, and this descent into hyperpolarization sets the membrane state of cortical neurons at a point of particular receptivity. When the ripple arrives and the hippocampal signal reaches the cortex, the cortical cells are positioned, by the spindle's timing, to respond to that input in a way they might not be positioned to respond to at any other moment. The cortex, in effect, has been prepared. The spindle trough is the threshold. The ripple's content arrives at the moment the door is open. This relationship, spindle preceding ripple, by 50 to 150 milliseconds, was documented in human subjects using simultaneous depth electrode recordings from the hippocampus and cortical surface EEG during natural non-REM sleep. Depth electrodes placed in the hippocampus of patients already undergoing monitoring for neurological reasons allowed researchers to record hippocampal ripples directly, while surface EEG recorded the cortical spindle. The timing was consistent enough to establish a directional relationship. The cortical spindle comes first, and the hippocampal ripple follows. The cortex leads, the hippocampus responds. The entire architecture of this three-frequency coupling, slow oscillation, nesting spindle, nesting ripple, provides the precise synaptic timing conditions that Hebbian plasticity requires. Hebbian plasticity is the process by which connections between neurons that fire together are selectively strengthened. Neurons that fire in near simultaneous coordination become over time more efficiently linked. The synapse between them, conducting the signal more reliably, the postsynaptic cell becoming more sensitive to input from the presynaptic cell. The critical word is near simultaneous. The window for this form of strengthening is narrow. Two neurons firing with too large a gap between them do not strengthen their connection. The gap must be small enough that both cells are active in the same brief window of molecular change. The three-frequency coupling creates that window, repeatedly, throughout every NREM episode. Each slow oscillation up phase brings the cortex into a state of widespread depolarization. Each spindle, nested in that up phase, drives rhythmic bursts of activity through thalamocortical circuits. Each ripple, nested in the spindle, delivers the compressed hippocampal replay to cortical circuits that are already active from the spindle's drive. Hippocampal input and cortical activity arrive together. The conditions for co-activation are met, and across hippocampal neocortical synapses, the long-range connections between the hippocampus and the distributed cortical regions that hold the semantic, perceptual, and contextual elements of a memory, the conditions for strengthening are fulfilled again and again, many times each NREM episode, dozens or hundreds of times each night. What passes through those synapses during replay is not a perfect recording. It is not a video playing back in a circuit. The hippocampus does not transmit the experience itself. It transmits the structure of the experience, the sequence, the temporal relationships, the linkages that bind one element of a memory to the next. The rat's place cell replay does not restore the maze. It restores the order in which positions in the maze were connected, and by restoring that order across the hippocampal neocortical pathway, it gradually allows the cortex to hold those connections on its own. This is what memory consolidation means at the systems level. The cortex, over many nights, absorbs what the hippocampus has been carrying until the hippocampus is no longer the only place where the memory lives. The spindle's contribution to this process is not merely to provide a timing signal. The spindle also opens a molecular pathway in the cortical neurons it drives. Calcium is the key that opens that pathway. In the apical dendrites of cortical pyramidal neurons, the long branching extensions that reach up toward the surface and gather input from many sources, the rhythmic spindle frequency depolarizations that arrive during the NREM up phase open a class of calcium channels called L-type channels, also known as voltage-gated calcium channels. These channels respond to the persistent rhythmic depolarization of spindle activity by emitting a brief inward surge of calcium ions. The calcium does not stay long. It diffuses inward from the channel and disperses within seconds. But in the moment of its presence, it acts as a molecular signal. That signal activates a protein called CAMP2, calcium calmodulin dependent protein kinase 2, which is among the most sensitive molecular detectors the synapse has of strong coincident activity. When calcium arrives, CAM2 activates. When CAM2 activates, it begins a cascade. It phosphorylates, or chemically tags, receptor proteins and scaffolding proteins at the synapse, making the postsynaptic density more receptive to subsequent input. It initiates the early phases of long-term potentiation, the synaptic process by which a connection, once strongly activated, becomes more efficient and more sensitive. This early phase is rapid but temporary. It can fade in a matter of hours if nothing follows it. What stabilizes it is protein synthesis, the production of new molecular machinery that physically reinforces the change. The protein synthesis is not triggered by the spindle alone, but the spindle's calcium signal may be exactly what the synapse needs to capture a protein synthesis wave when one becomes available. This is the synaptic tagging and capture hypothesis. The idea is that when a synapse is strongly activated during waking, during the learning of something new, it acquires a transient molecular tag. The tag is placed by the early phase LTP processes activated during the waking event. It does not immediately change the synapse in any permanent way. It marks it. It marks it as a synapse that should be strengthened if the resources to do so become available. The tag remains for several hours, and then, during subsequent sleep, spindle-driven activity provides calcium, which provides CAMP2 activation, which drives protein synthesis in the activated neurons. That protein synthesis wave reaches all synapses that are tagged, the ones marked during waking learning, and at those synapses specifically, the early phase change is captured, stabilized, and converted into late phase long-term potentiation. The connection becomes permanently stronger. The memory in the molecular language of the synapse has been preserved. The elegance of this mechanism lies in its selectivity. Spindle-driven calcium and protein synthesis do not strengthen every synapse in the cortex. They strengthen the ones that were tagged, and the tags were placed by waking experience. The sleeping brain is not strengthening synapses at random. It is completing a process that waking learning began, using the molecular marks left by the day's events as the guide to where the night's consolidation work should be done. The result is that sleep strengthens what was learned rather than every trace of activity the brain ever encountered. Deep in the sleeping brain, in the curved fold of tissue that curves from the temporal lobe inward toward the center, the hippocampus holds its replays in brief bursts, each one shorter than the blink of an eye, each one delivering a compressed trace of a waking experience to the cortex above and around it. The ripples fire, the cortex receives them, and across the long, slow arc of an non-REM episode, across 4 or 5 or 6 episodes through the night, the accumulated weight of many thousands of small replays gradually shifts the distribution of synaptic strength in the cortical networks that will carry the memory from that night forward. A word and its pair sit in a memory trace somewhere in the temporal cortex, connected by a link that was new and fragile when the day ended. By morning, if sleep has been uninterrupted and the non-REM episodes have been long and the spindle activity has been sufficient, that link is stronger. The word arrives and the pair follows. Not because the brain was rehearsing, not because there was any conscious repetition in the night, but because in the dark and in the quiet, while awareness was suspended, the oscillations that organize sleep were also organizing memory. This was demonstrated in a laboratory setting with elegant simplicity. Participants learned pairs of words before sleep, arbitrary pairings, the kind that require effortful learning, because the words themselves suggest no natural connection. They were tested on the pairs the morning after sleeping, and the participants who showed the highest spindle density in the non-REM sleep of the intervening night remembered the most word pairs the next morning, not the participants who slept longest, not the participants who had the most total sleep time, the participants with the most spindles. The detail that makes this finding precise is what spindle density did not predict. Procedural memory, the kind of motor learning involved in tracing a mirror image or learning the sequence of keystrokes in a new skill, did not correlate with spindle density in the same way. Spindles appeared selectively beneficial for declarative memory, the memory of facts and events, the kind of memory that requires the hippocampus during its formation, the kind of memory that benefits from the hippocampal neocortical transfer that the 3-frequency coupling is believed to accomplish. The spindle's role was not general, it was specific, and the specificity matched the mechanism. Spindles at the cortical surface, ripples in the hippocampus, and the coupling between them driving a transfer that is relevant precisely for the memories that the hippocampus initially holds. Jan Born's group at the University of Tübingen produced this finding, and it stands as one of the clearest behavioral demonstrations that sleep spindle activity is not simply a correlate of light sleep, not simply a marker of sleep quality, but a mechanistic contributor to the process by which the sleeping brain converts recent learning into durable memory. The correlation between spindle density and word pair retention was not mediated by total sleep time, or by sleep efficiency, or by the duration of stage 2 NREM sleep alone. The spindle density itself was the predictor. To sit with this finding for a moment is to arrive at something quiet and particular. A person sleeps. They are not aware of anything. The oscillations that characterize NREM sleep move through their brain in the same broad patterns they moved through 10,000 years ago and will move through 10,000 years from now. The slow oscillation rolls. The spindles nest within it. The hippocampal ripples nest within the spindles. And in the brief windows those nested rhythms create, the synapses that hold what was learned that day receive the molecular signals that will make them hold it longer. The person wakes, and the words are there. They do not know why. The mechanism is invisible, and the experience of remembering feels simple. The word arrives, the pair follows, and there is no trace left in waking awareness of the process that preserved it. What makes the three-frequency coupling important is not just the elegance of its architecture. It is the precision of its timing. The spindle trough preceding the ripple by 50 to 150 milliseconds is not an approximate relationship. It is a specific one. In simultaneous recordings of hippocampal depth electrodes and cortical EEG in human subjects, the timing of spindle phase relative to ripple onset was consistent enough to suggest a functional relationship rather than coincidence. The cortex, through its spindle, actively gating the conditions under which the hippocampus replays. The direction of the relationship runs from cortex to hippocampus. When the cortical spindle is absent, the probability of a hippocampal ripple is lower. When the spindle is present, the ripple is more likely to occur at a specific spindle phase, and more likely to produce the correlated activity in cortical neurons that Hebbian plasticity requires. The cortex, in other words, does not simply receive what the hippocampus sends. It creates the conditions under which the hippocampus sends it. The spindle is not a passive recipient of hippocampal replay. The spindle is part of the circuit that enables replay to happen at all. This bidirectionality, hippocampus and cortex in conversation, each shaping the other's state across the span of a non-REM episode, is one of the places where the systems level and the cellular level descriptions of consolidation converge. The coupling hierarchy that permits Hebbian plasticity at the synapse is the same hierarchy that organizes the replay events that deliver the information those synapses need to receive. A cortical pyramidal neuron rests in the deep layers of the temporal cortex, its long apical dendrite reaching upward through 4 or 5 layers of neural tissue toward the cortical surface. It has been active today. A word pair entered through auditory cortex and reached the hippocampus, where it was bound into a trace by the associative machinery of the hippocampal formation. Some signal of that binding returned to this neuron, leaving in it the chemical marks of early phase activation, a slight shift in the receptor composition of its dendritic spines, a kinase held in a partially activated state, a tag on the machinery of the synapse. The neuron is quiet now. In the first NREM episode after learning, the slow oscillation rolls over it. A spindle arrives on the slow oscillation's up phase, and the spindle's rhythmic depolarizations open the L-type calcium channels in the apical dendrite. Calcium enters. CAM2 activates. Protein synthesis begins, targeted at the tagged synapses. The hippocampus sends a ripple, and the signal it carries arrives at exactly the moment the calcium-driven plasticity cascade is running. The combination, the replay signal and the calcium-initiated plasticity machinery running simultaneously creates conditions under which the synaptic connection encoding the word pair is strengthened in a way it would not have been strengthened by either process alone. This is not a metaphor. The calcium is real calcium. The protein synthesis is real protein synthesis. The synapse that strengthens is a physical structure, a density of receptor proteins and scaffolding proteins and signaling molecules embedded in the membrane of a dendritic spine, and its change from that night forward is a physical change, measurable with enough precision, stable enough to persist for days or weeks or years. The word pair is now held not only by the hippocampus, but by the cortical synapse itself. The connection between the representations of the two words, reinforced at the cellular level by a molecular process that was set in motion by an oscillation the sleeping person never knew was there. The slow oscillation continues to roll through the cortex. Each up phase brings another window, and in that window another spindle, and inside the spindle another ripple, another brief replay, another calcium transient diffusing inward through another dendritic spine. The process repeats, not once or twice across the night, but dozens of times per NREM episode, across 4 or 5 or 6 NREM episodes, each episode adding its accumulated weight of replays and plasticity events to the consolidation that has been accumulating since the day's learning ended. There is something about the scale of this that resists easy description. Each individual event, each ripple, each calcium transient, each brief activation of CAMP2 at a tagged synapse is vanishingly small. The ripple lasts 1/10 of a second. The calcium transient diffuses across a space smaller than the width of a hair. The protein synthesis, triggered at a single synapse, is invisible to any imaging instrument currently available for use in a living brain. And yet across the course of a night, these events accumulate into something measurable at the behavioral level. Words remembered, facts held, maps preserved. The smallest molecular events of the night translate, through sheer repetition and through the precise organization of the oscillatory coupling that orchestrates them, into the capacity to wake and know what was known the evening before, and more. The 3 rhythms are nested so that the fastest can act inside the window, the middle one creates inside the window, the slowest one provides. Each rhythm is a different temporal scale of the same conversation. The slow oscillation is the background of the night itself, the broad shifting of the cortical membrane that distinguishes the sleeping brain from the waking one. The spindle is the intermediate rhythm, brief and recurrent, the deoscillation that opens the calcium channels and times the cortical reception of hippocampal input. The ripple is the content, the compressed, ordered sequence of a waking experience replayed with the precision of a circuit rather than the loose reconstruction of memory. No single rhythm does the work alone. A cortex with spindles but no ripples has the timing without the content. A hippocampus with ripples but no spindles, delivering the correct membrane conditions, has the content without the receptive context. A brain with spindles and ripples, but no slow oscillation to organize their placement within the up phase, has the events without the ordering architecture that determines which events reach cortical synapses in their most plastic state. The 3-frequency coupling is the mechanism because all 3 frequencies are required. Their nesting is not incidental. It is the structure that makes the transfer work. In the CA3 region of the hippocampus, the sharp wave begins. It is a large, slow voltage shift, the product of recurrent excitatory connections between CA3 pyramidal cells that have been active since the learning event earlier in the day. CA3 is a region of dense interconnection. cells talking back to each other in loops that can sustain and amplify patterns of activity. When the brain is in the correct NREM state, a pattern that was active during waking can reemerge spontaneously in CA3, and the sharp wave it generates propagates through the trisynaptic circuit to CA1, where it drives the coordinated burst firing of the ripple. The ripple in CA1 is the output signal. It travels down the hippocampal output pathways toward the cortex, and it carries in the timing of its firing the compressed sequential information that the CA3 pattern contained. That information reaches the cortex as an arriving excitatory signal at the precise synaptic terminals that were active during the original learning event. It does not arrive at random cortical synapses. It arrives at the synapses that hold representations connected to the experience that was replayed. And at those synapses, if the spindle has delivered its calcium, if the plasticity machinery is running, if the timing alignment of the three nested rhythms has placed the ripple at the correct moment of the cortical membrane cycle, the arriving signal finds a receptive plastic synapse and the connection is strengthened. Outside the skull, in the cool quiet of a sleeping room, none of this is audible or visible. The breath rises and falls. The body is still. The EEG electrodes, if they are there, show the slow rolling background of NREM sleep, punctuated by the brief sigma band bursts of spindles, a waxing and waning cluster of waves at 11 to 15 cycles per second, rising over half a second, cresting, falling back into the flat background silence. Each of those bursts is a window opening. Each window opening is an opportunity. And over the long arc of the night, from the first NREM episode through the last, the windows open and close, the ripples fire inside them, and the molecular work of memory consolidation proceeds without any participation from the sleeping mind. A word pair learned in the afternoon sits in the hippocampus when the body lies down. The sleep comes, and the first slow oscillations sweep through the cortex, and the first spindles nest within them, and the hippocampus begins its replays in brief, precise bursts, and the molecular machinery at the tagged synapses begins its quiet work. This happens for 6 or 7 or 8 hours, and when it ends, when the last NREM episode releases into the early morning sleep that is lighter and less rich in slow oscillation, and the slow oscillations thin, and the spindle density shifts, and the night moves toward morning, the hippocampus holds a slightly lighter load than it did when the night began. Some of what it was carrying has been transferred. The cortex holds a little more than it did before. The words remembered the next morning are the measure of all that invisible work, not a complex measure, not a measure that requires understanding anything about oscillations or ripples or calcium or protein synthesis. Just the words, arriving when called for, as though they had always been there, which by morning, in the small local sense of a strengthened synapse, they very nearly have. The hippocampus sent them into the cortex across the quiet hours of the night. The spindle opened the door each time, and when the door closed for the last time before waking, the words were on the other side of it, settled into the synaptic architecture of the cortex, waiting in the slow, permanent silence of a strengthened connection for the moment they would be needed. That is the consolidation conversation. It has no sound. It leaves no trace in the morning awareness of the person in whom it happened. It requires nothing of the sleeper except the sleep itself, the slow oscillations coming, the spindles nesting within them, the hippocampus sending its brief compressed ripples through the narrow windows that the spindle timing provides. The three rhythms turn through the night together, nested and precise, each one doing what the others require of it, and by the time the light comes and the eyes open, and the question arrives, what was the word, what was the pair, What was it that needed to be remembered? The answer is already there, built in the dark from the small and silent currency of calcium and protein, and a tenth of a second of coordinated firing, repeated patient and unhurried through the long quiet hours of the sleeping night. Two polysomnography records rest side by side on the same screen, their lines scrolling through the quiet of a sleep laboratory sometime after midnight. The room is dark except for the pale glow of the monitor. Both records belong to healthy adults sleeping in adjacent rooms, each one breathing slowly, each one several hours into a night of undisturbed sleep. But the records do not look the same. One shows the sigma band, alive with dense repeating bursts, tight clusters of waves rising and falling every few seconds across each NREM episode. The spindle activity almost continuous in places, a kind of electrical abundance that fills the trace. The other is quieter. The sigma band is still present, the spindles still there, but they arrive with more space between them, fewer per minute, smaller in their amplitude, briefer in their duration. Both sleepers are resting, both are unaware of any difference. The difference lives entirely in the oscillatory architecture of the night, invisible to the people who carry it, legible only to the instruments recording it. This is where the science of sleep spindles becomes personal, in the quietest possible way, not dramatic or troubling, just the fact that the sleeping brain is not uniform across people, that some of its most important nighttime activity varies from one individual to the next in patterns that are stable, partially inherited, and quietly consequential for what the morning holds. The spindle-dense sleeper and the spindle-sparse sleeper will wake at roughly the same time. They will feel, in many cases, comparably rested. But if both of them had spent the previous afternoon learning a list of word pairs, or committing the layout of a new building to memory, or encoding the subtle sequence of a skill, the sleeper whose night held more spindles will, on average, remember more. The difference is not absolute. It is not destiny. It is a statistical tendency measured across groups, reliable enough to appear in study after study, gentle enough that no single night tells the whole story. And it begins not in the morning, not in the testing room, but in those quiet hours between when the light went out and when it came back on. Matthew Walker's group at the University of California, Berkeley, documented this clearly. Participants who showed higher spindle density across an overnight sleep period retained significantly more declarative memory, word associations, spatial arrangements, factual connections than participants matched for total sleep time, but lower in spindle counts. Total sleep time was held constant. The group slept the same number of hours. The difference was in the texture of what happened during those hours, specifically in how often and how richly the thalamocortical circuit produced its brief, self-contained oscillatory bursts. More spindles across the night meant more remembered in the morning. Fewer spindles meant less. The nights looked similar from the outside. The EEG told another story. What makes this finding hold its weight is the specificity of it. Sleep does many things. It restores metabolic balance. It clears cellular waste. It regulates emotion and refreshes attention. Many of these restorative processes scale with total sleep time, with sleep pressure, with how much slow wave activity accumulated before dawn. But spindle density, as a predictor of declarative memory retention, goes beyond what total sleep time explains. When the statistics account for how long someone slept, spindle density still predicts what they remember. The spindles are not simply a byproduct of getting enough sleep. They are doing something particular, something that belongs specifically to them. The question that follows, why some sleeping brains produce more spindles than others, turns out to have a deep and partly genetic answer. The thalamocortical circuit that generates spindles is not built identically in every person. Its components, the inhibitory neurons of the thalamic shell, the relay cells they drive, the calcium channels that allow the rebound bursting, the GABA receptors that set the depth and duration of the inhibitory pause. All of these are built from proteins encoded by genes that vary across the human population. That variation is not random noise. It is heritable. It passes from parent to child in patterns that twin studies can detect. The Borbeli group in Switzerland, working with identical and fraternal twin pairs, found that spindle characteristics, frequency, density, amplitude, the overall power in the sigma band, show high heritability. The estimates range from roughly 44% to over 80%, depending on the specific characteristic and the specific study. Identical twins whose genomes are copies of each other produce spindle records that are far more similar than those of fraternal twins who share only half their genetic material on average. The frequency at which the thalamus oscillates during sleep, the density of spindle events per minute, the amplitude of each burst, these are not arbitrary. They reflect a circuit architecture that is substantially determined, though not fully determined, by the genetic blueprint the person was born with. Genome-wide association work has pointed towards some of the variants involved. Genes encoding calcium channel subunits, the proteins that make up the low threshold channels in thalamic neurons that allow rebound bursting, carry variants that predict spindle characteristics. Variants in CACNA1C and CACNA1H, two genes involved in the construction of voltage-gated calcium channels, have been implicated. So have variants in genes encoding GABA receptor subunits, including GABRA1, which shapes the duration and depth of the inhibitory signals that set the interburst interval and thus the frequency of the oscillation. These are not large effects. Each individual variant contributes a small fraction of the variance, but together, across many genes, they build a thalamocortical circuit that produces a characteristic spindle signature, one that is partially predictable from the genome and substantially stable across time. That stability across time is itself a notable feature. Sleep spindle characteristics, unlike many physiological measures that shift with stress or illness or the hour of the night, show moderate to high consistency across nights separated by weeks or months. The same person sleeping in the same laboratory on 2 different nights, separated by a month of ordinary life, produces spindle records that resemble each other more than they resemble the records of different individuals. Spindle density, frequency, and topography are in this sense closer to a trait than to a state. They are more like blood type or bone density than like heart rate or cortisol, not entirely fixed, but far less fluid than moment-to-moment measures of arousal or metabolic activity. This is what gives the Walker group's finding its staying power. If spindle density were a nightly fluctuation, correlated with memory performance on a given night, by chance or by some third variable, the correlation would be noisier and harder to interpret. The trait-like stability means that high spindle individuals are consistently high spindle, and their consistently better declarative memory retention is not a lucky coincidence of a single good night. The genetic architecture of the spindle generating circuit also raises a question that the data has begun to answer, though not yet fully, if spindle characteristics are heritable, and if they predict memory consolidation. Does spindle density also predict broader cognitive performance? The evidence suggests that it does, at least in part. Robert Bodies and colleagues found that parietal fast spindle amplitude, the size of the high-frequency spindle bursts, measured over the rear of the scalp, was the strongest EEG predictor of full-scale intelligence scores across a diverse group of participants. The relationship held after controlling for sex, age, and socioeconomic background. Participants with larger parietal fast spindles scored higher on standardized cognitive tests. The effect was not enormous, but it was consistent and specific, specific to the fast spindle band, specific to the parietal region, and independent of the confounds that typically complicate this kind of analysis. The proposed mechanism behind this relationship does not require anything exotic. The same thalamocortical circuit properties that produce large, dense, precisely timed spindle bursts during sleep, efficient inhibitory control, precise rebound timing, reliable propagation from thalamus to cortex, are likely to be the same properties that support rapid, precise information processing during waking. The thalamocortical relay is not only a sleep structure, it is the primary pathway through which sensory information reaches the cortex during waking hours, filtered and gated by the same inhibitory mechanisms that produce spindles in sleep. A circuit that performs this function with precision during the day may produce cleaner, better timed spindle bursts at night. A circuit that produces cleaner spindles at night is by that same token a circuit that handles information efficiently during the day. The sleep signature and the waking cognitive performance may not be two separate things connected by some invisible link. They may be two visible expressions of the same underlying circuit quality. Still, the relationship between spindle density and intelligence is not a closed book. The correlation is reliable but modest. Many high-intelligence individuals produce unremarkable spindle records, and some individuals with moderate cognitive test scores show rich spindle activity. The relationship is real enough to study and too noisy to use as a predictor for any individual person. What the data establishes is a directional tendency, not a rule. One more thread in a pattern that grows clearer the more it is examined from different angles. Age adds another thread, and it is one of the quieter findings in the field. Sleep spindles change across the lifespan in a direction that is not reversible, and the change begins well before old age. Through childhood and early adolescence, spindle density tends to be high. The thalamocortical circuit is actively developing during these years, and the sleep of childhood contains more slow-wave activity, more coupling between slow oscillations and spindle bursts, and generally richer sigma band power than adult sleep. Then, beginning somewhere in the fourth and fifth decades of life, a range, not a sharp threshold, spindle characteristics begin to decline, density falls, amplitude shrinks. The durations of individual events grow briefer. The neuroanatomical basis of this decline has been traced, at least in part, to the thalamic reticular nucleus itself. Post-mortem histological studies and diffusion tensor imaging of living participants show that the thalamus loses volume with age, and that the white matter tracks connecting thalamic relay neurons to the cortex become less structurally coherent over decades. The fibers thin. Their capacity to conduct precise, synchronized oscillatory signals from thalamus to cortex diminishes. What was once a reliable high-amplitude burst arriving simultaneously across a broad cortical area becomes something smaller, slower to propagate, less synchronized. The spindle that an EEG electrode at the crown of the scalp records in a 50-year-old is on average quieter than the one recorded from the same electrode position in a 25-year-old, not because the brain is less active in total, but because the specific circuit that generates sigma band bursts has undergone a structural change that no amount of sleep extension fully compensates for. There is something slow and inevitable about this, described clearly and without alarm, the same kind of change that thins the lens of the eye or reduces the peak force the heart can generate, a settling of biological machinery that has been running continuously for decades. It does not mean that memory consolidation ceases in middle age. Sleep continues to strengthen memory throughout life. The mechanisms remain present, but the efficiency of the consolidation engine changes, and the spindle record documents that change in a way that is visible, measurable, and tied to real differences in overnight memory performance across age groups. None of this is fixed. The circuit can be engaged and its activity modulated, though the range of influence is bounded. The spindle generating architecture is a biological given, shaped by genes and by the long structural changes of aging, but it is also embedded in a body whose sleep is affected by stress, by caffeine, by the timing of the sleep period, by light exposure in the hours before bed, by alcohol and sedatives, and the thousand variables of ordinary life. Some of these variables suppress spindle activity. Alcohol reliably reduces spindle density in the first half of the night. Some anxiolytics and many common sleep aids alter spindle characteristics in ways that depend on which receptor subtypes they engage. The circuit is not mechanical in the sense of being immune to influence. It is biological, which means it lives in a context and responds to it. In the other direction, toward more spindle activity, more coupling, more efficient consolidation, the science has become increasingly precise, though the story of intervention belongs elsewhere. What matters here is simply the baseline, that the spindle density a person produces on a typical night of undisturbed sleep reflects something real and stable about the circuit they carry, and that circuit's activity across those quiet hours shapes, in ways that are reproducible and measurable, what they will be able to retrieve the next day. The most vivid illustration of the high end of spindle biology comes not from comparing averages across large populations, but from looking at individuals whose memory abilities sit well outside the ordinary range. There is a group of people, small in number, who possess what researchers have called highly superior autobiographical memory, the capacity to retrieve the details of almost any day of their lives with a precision and completeness that most people cannot approach. These individuals can describe what they were wearing, what they ate, who they spoke to, what was in the news, on almost any given date, going back decades. Their autobiographical recall does not fade and compress the way ordinary memory does. It remains detailed, accessible, and apparently complete across long stretches of time. When these individuals undergo polysomnography, their sleep records are different from controls. The difference is not in total sleep time, which is unremarkable. It is not in the proportion of the night spent in each sleep stage, which is also broadly normal. The difference is in the spindle activity. They show elevated spindle density relative to matched controls, more events per NREM episode, more sigma band power across the night. Their spindle topography is slightly altered. The sigma band activity tends to persist longer within individual episodes and to extend across a broader reach of the scalp than controls show, as if the thalamocortical conversation continues a beat longer on each cycle, covering more of the cortical surface before it quiets, and the coupling between spindle events and hippocampal sharp wave ripples, the precise temporal linkage between the thalamic oscillation and the hippocampal replay, occurs more frequently per NREM episode than in controls. This last point carries particular weight. The coupling between spindles and sharp wave ripples is not just a measure of how many spindles occur. It is a measure of how effectively the spindle generating circuit is coordinating with the hippocampal replay machinery. How often the timing of a cortical spindle burst coincides precisely with the compression and transmission of a hippocampal memory trace. More coupling events per NREM episode means more opportunities per night for the hippocampus to send its replayed sequences to the cortex under the ideal synaptic conditions that a spindle burst provides. It means more transfers completed, more traces strengthened, more of the day's experience secured into the slower, more stable form that survives the weeks and months that follow. For individuals with highly superior autobiographical memory, this elevated coupling suggests that their extraordinary retention may not be purely a matter of how they encode information during waking hours, though that may also differ. It may be substantially a matter of what their sleeping brains do afterward. The consolidation engine runs more efficiently through the night. The transfers happen more often and perhaps more completely. By morning, more has been moved from the fragile temporary store to the durable long-term form that will still be accessible decades later, and the EEG record of those sleeping hours carries the signature of a circuit that was doing this work with unusual thoroughness. There is something worth sitting with in this, the aspects of memory that are most admired, recall of fine detail, the capacity to retrieve experiences from the deep past, the richness and completeness of what is preserved. These tend to be attributed, in everyday understanding, to something that happened during the experience itself. How attentive the person was, how emotionally engaged, how deeply they processed what was in front of them. And those factors do matter. Encoding has real consequences for what is available to be consolidated, but the data from highly superior autobiographical memory suggests that some of the most important work is done in the hours after the experience ends, in a sleeping brain that the person is not aware of, in a circuit that fires its brief rhythmic bursts, a few hundred times more per night than the average person's does. The memory achieves its permanence not only at the moment of experience, but in the quiet that follows, in the spindle-rich oscillatory architecture of a particular kind of night. This picture of memory ability as partly a nighttime biological trait, rather than only a waking cognitive one, is new enough that its full shape is still being mapped. The highly superior autobiographical memory findings come from small samples, and replication in larger groups is ongoing. The direction of causation is not always easy to establish. Does the elevated spindle activity produce the superior memory, or does a brain with different cortical architecture both encode more richly during waking and generate more spindles at night, the two being parallel expressions of the same underlying difference? Both may be true. They are not mutually exclusive. What the data establishes is a reliable association between spindle biology and memory ability at the high end of the distribution, consistent with the same relationship found across more typical ranges of performance. Also consistent with the participant-level selectivity finding that individuals with higher performance on a particular memory task, the mnemonic similarity task, which tests the precision of episodic memory encoding, pure show altered spindle characteristics compared to matched controls, more spindle events, different topography, more coupling. The relationship between spindle biology and memory ability appears across multiple memory domains, multiple measurement approaches, and multiple points along the distribution from ordinary to extraordinary. What this adds up to, for the person lying in a sleep laboratory with 19 electrodes on their scalp, or for anyone whose brain is doing this same work on any ordinary night, is a portrait of sleep as something more structured and more individually characterized than the uniform rest it appears to be from the outside. The sigma band activity of each NREM episode, those brief, rhythmic, self-contained bursts that rise in amplitude for half a second, crest, and then taper back into the slower background, is not the same for every sleeper. It is shaped by the genes that built the circuit, by the decades of structural change the thalamus has undergone, by the night's particular conditions, and by something that seems to reflect, in its efficiency and its coupling to hippocampal replay, the memory capacity that will be available the next morning and the year after that and the decades to come. The sleeper with the denser, richer spindle record does not know what the night is doing on their behalf. They are not directing it. They are not aware of the rebound bursts in the thalamic shell, the synchronized propagation up to the cortex, the precise temporal coincidence between a cortical oscillation and a hippocampal replay event. They are simply asleep. The brain is simply doing what brains do during non-REM sleep in the particular way that this brain does it, with the particular circuit architecture it happens to have inherited and maintained and carried into this night. But the morning will know. The morning always knows. Not in a dramatic way, not in a sudden clarity or a conscious recognition that something was secured during the night. It will show up in the quiet reliability of recall, a word pair retrieved cleanly, a spatial arrangement remembered precisely, a detail from last Tuesday still accessible without effort, still present and usable, having survived the night in the form in which it was learned. That survival is not passive. It is the accumulated result of the spindle activity that ran through each non-REM episode from the first hour of sleep to the last, brief, precise, rhythmic, invisible, doing the particular work that only this oscillation does. Two records on a screen, side by side, in a dark laboratory, one dense with sigma band activity, one quieter, both sleepers breathing slowly, equally at rest, equally unaware. The difference between them is encoded in the EEG, in the character of the bursts, in how many times per non-REM episode the circuit completed its cycle and sent another wave of synchronized depolarization from thalamus to cortex and back again. It is a difference measured in counts per minute, in microvolts of amplitude, in the precise millisecond timing of coupling events that happen too quickly and too far below awareness for either sleeper to sense. By the time they wake, the difference will have expressed itself in something far simpler, in what each of them still knows, and in how much of yesterday and of the years before yesterday is still quietly and reliably there, held in place by the oscillatory work of the night, in those brief repeating bursts. In the slow, rich dark, 19 electrodes rest against a sleeping scalp in a quiet room. The room is dark. The instruments around the bed make no sound beyond the faint, continuous hum of cooling fans, and the only light comes from a monitor on the far wall, where a long green line traces the slow electrical weather of a brain that has been asleep for nearly 2 hours. The line does not move like a heartbeat, with its sharp, familiar spike. It moves the way a tide moves, broad, unhurried, one great swell rising and subsiding over the span of several seconds, carrying within it smaller rhythms, too fine to see unless the display is stretched. The researcher sitting at the monitor is not watching for the slow waves. Those are visible without effort, their amplitude so large they dominate the trace. What she is watching for is something smaller, nested inside the slow wave's rising phase, a brief cluster of faster oscillations that appear for half a second and then fade, leaving the trace as quiet as it was before. When it comes without announcement. The line tightens slightly, waxes through 5 or 6 quick cycles, reaches a modest crest, and then tapers back into the broad rolling background. It lasts less than 2 seconds, then it is gone. That brief cluster has a name. It is a sleep spindle, and the sleeping brain in that room has already produced several hundred of them tonight, one by one, in the dark and silence of each NREM cycle, while the person to whom the brain belongs remains entirely unaware. The full record of a night's sleep, printed out at clinical scale, runs to more than 1,000 pages. Most of it is background, the slow oscillations of deep NREM, the jagged irregularity of REM, the flat stretches of lighter sleep where the brain idles near waking. The spindles are brief punctuations scattered through that long record. Each one is narrow, compact, lasting between half a second and 3 seconds, never longer. Each one sits in the sigma frequency band, between 11 and 16 cycles per second, fast enough to look like a tight tremor on the trace, slow enough that each individual wave is distinct and countable. Between the spindles, the trace returns to its unhurried baseline, as if nothing happened, but something did happen. And what happened during each of those brief bursts is the question that has occupied sleep researchers for several decades, carried through long nights in laboratories on multiple continents, generating a body of evidence careful and cumulative enough to support a specific answer. Spindles are not background noise. They are not artifacts of the sleeping brain, idling between one phase and the next. They are the mechanism by which the sleeping brain selectively preserves what was worth learning during the hours before. The polysomnography setup, the full arrangement of electrodes, amplifiers, and recording equipment, remains the most precise tool available for studying spindle activity in sleeping humans. The 19 scalp electrodes, distributed according to the International 1020 system, each pick up the aggregate electrical signal of millions of neurons cycling below. The central electrodes, positioned roughly above the midline of the scalp, register the strongest spindle signal because it is from the thalamus, deep below and slightly forward of center, that the rhythm originates and fans upward through thalamocortical circuits to the cortex. The electrodes at the back, over the parietal regions, capture a somewhat different spindle character, faster, more locally organized, distinct from the slower frontal spindles that dominate the forward channels. A trained scorer working through a night's record by eye can identify spindles reliably, but not perfectly. Two expert scorers examining the same record will agree on a large proportion of spindle events, but not all of them. The boundary between a true spindle and a brief run of sigma frequency noise is not always sharp. When agreement between experts is measured formally as an intraclass correlation coefficient, it typically falls between about 7/10 and 8.5 high, but not absolute. This is not a failure of the method. It reflects the genuine ambiguity at the edges of a biological signal that grades continuously into the surrounding background rather than switching cleanly on and off. The ambiguity has been one reason automated detection methods have grown steadily more capable and more widely used. The basic approach is straightforward. Bandpass filter the EEG signal to isolate the sigma band, calculate a running measure of amplitude, and flag any interval where that amplitude exceeds a threshold, typically set at 1.5 to 2 standard deviations above the mean signal, for a duration of at least half a second. This captures most spindles. It misses some. It occasionally flags non-spindle activity. The threshold is a practical compromise between sensitivity and specificity, and different laboratories have historically set it in slightly different ways, which is one of the less discussed reasons why spindle counts from different studies are not always directly comparable. Convolutional neural networks trained on large polysomnography databases have pushed automated detection further, trained on the sleep EDF dataset and others compiled from multiple sites. These networks have reached F1 scores, a measure that balances the cost of missed detections against the cost of false ones, exceeding 8/10, a level approaching the agreement between two human experts scoring the same data independently. The advantage over the threshold method is not only accuracy, it is the richness of the output. A neural network working through a night of multi-channel EEG can return not just a list of spindle timestamps, but a full parameter matrix, the onset and offset of each event, its peak frequency within the sigma band, its duration, the rise time and fall time of its amplitude envelope, its spatial distribution across the scalp, and a measure of how symmetrical the waxing and waning shape is. Assembling that parameter matrix by hand for a single night of sleep would take a skilled researcher many hours. The network produces it in seconds. That richness matters because the spindle is not a single uniform event. Fast spindles and slow spindles differ in their topography and their behavioral correlates. Fast spindles, those peaking above about 13 cycles per second, are strongest over the parietal regions of the scalp, and it is fast parietal spindles that have shown the most consistent associations with declarative memory performance. Slow spindles, peaking near 11 or 12 cycles per second, tend to predominate over frontal regions and have been more variably linked to memory outcomes across studies. The distinction is not purely academic. Treating all spindles as functionally interchangeable would be like treating all precipitation as the same because it falls from the sky. The parameter matters for the outcome. Across the body of research into sleep spindle function, one behavioral result has been replicated with unusual consistency. The number of spindles a person generates during the non-REM sleep following a period of learning predicts how much of that learned material they will retain by the following morning. The relationship holds for word pairs, for spatial location tasks, for associative memory tasks that require linking one item to another. It holds after controlling for total sleep time, for slow wave sleep duration, and for time in bed. Participants who generate more spindles retain more. Participants who generate fewer retain less. The correlation is not perfect. Memory consolidation draws on multiple processes during sleep, and spindles are not the only contributor, but it is consistent enough across enough laboratories and enough subject populations to be taken seriously. The causal direction of this relationship, whether more spindles produce better memory, or whether better learning before sleep triggers more spindles, or both, has been one of the central questions driving the experimental branch of spindle research. Correlational studies, however careful and however large, cannot answer it. Answering it requires either selectively enhancing spindle activity and measuring memory, or selectively disrupting spindles and measuring the cost. Both approaches have been tried. In 2013, a research group working at the University of Lübeck published results from a closed-loop stimulation experiment that has since become one of the most cited studies in sleep and memory research. Their method depended on detecting the slow oscillations of non-REM sleep in real time, catching each rising phase as it unfolded, within milliseconds of its onset, and delivering a brief tone precisely into that rising phase, quiet enough not to rouse the sleeper, timed precisely enough to interact with the oscillatory circuit already underway. The slow oscillations upstate, the depolarized phase when the cortex is briefly active before falling silent again, is the window during which spindles nest most reliably. By reinforcing that upstate with a precisely timed sound, the stimulation nudged the oscillatory circuit toward a slightly larger amplitude cycle, one that generated more spindle activity nested within it. The memory test was simple. Before sleep, Participants learned lists of word pairs. After sleep, they were tested on how many pairs they could recall. The participants who received the closed-loop stimulation during NREM sleep recalled roughly 17% more pairs the following morning than participants who slept undisturbed or who received tones delivered out of phase with the slow oscillation, a sham condition designed to control for the effect of sound alone. The 17% improvement was not explained by total sleep time, which was the same across groups. It was not explained by the tones themselves, which in the out-of-phase condition produced no benefit. The improvement came specifically from the timed interaction between the stimulation and the oscillatory architecture of NREM sleep, an interaction that boosted slow oscillation amplitude and, coupled within it, spindle density. That finding is worth pausing over. A sound, quiet enough that the sleeper heard nothing consciously, delivered with millisecond timing to the rising phase of a slow oscillation in a sleeping brain, produced a measurable improvement in next morning memory retention, not through any waking awareness, not through any encoding at the time of the sound, through the nudging of an oscillatory circuit already at work in the dark, doing what it does every night, slightly more effectively. The mechanism by which the stimulation worked connects back to the coupling hierarchy that underlies memory consolidation during NREM sleep. The slow oscillation organizes the cortex into alternating up states and down states. During the up state, the cortex is briefly active, and it is during this brief activity window that sleep spindles preferentially occur. Within the spindle itself, the rhythmic depolarization of thalamocortical relay neurons sends waves of excitation into the cortex at 11 to 15 cycles per second. And nested within the spindle, in precise temporal alignment with the spindle troughs, hippocampal sharp wave ripples replay compressed sequences of prior waking experience. The three rhythms are coupled. The slow oscillation sets the timing. The spindle gates the cortical window. The ripple delivers the hippocampal content. Boosting the slow oscillation boosted the spindle. Boosting the spindle strengthened the conditions under which hippocampal replay could be received and used by the cortex. The closed-loop approach did not invent this process. It leaned into it. It found the oscillation already underway, added just enough energy at the right moment, and let the brain's own architecture do the rest. The improvement in memory was not produced by any external encoding. It was produced by making the brain's own consolidation machinery run a little more efficiently for a few minutes in the dark. Pharmacological approaches to spindle enhancement offer a different angle on the same question. Zolpidem, a compound that increases GABA activity at specific receptor subtypes, reliably increases sigma band power during sleep. At therapeutic doses, zolpidem boosts spindle density, increases spindle amplitude, and prolongs spindle duration. In a study published in the same year as the closed-loop stimulation result, participants who took zolpidem before a daytime nap following motor sequence learning showed better procedural memory consolidation than participants who received a placebo. Their enhanced spindle density during the nap correlated directly with the degree of improvement. The pharmacological result is not as clean as the closed-loop result. Zolpidem's effects on the sleeping brain extend well beyond spindles. It alters REM sleep, suppresses slow-wave activity to some degree, and produces some degree of anterograde amnesia at higher doses. The specific attribution of the memory benefit to spindle enhancement rather than to other GABAergic changes cannot be made with full confidence from pharmacology alone. Tolerance develops with repeated use. The compound is not a selective spindle amplifier in the way that a precisely timed sound delivered to a rising slow oscillation is. Both strands of evidence, the stimulation and the pharmacology, point in the same direction. Neither is sufficient on its own to close the question entirely. What the two approaches together establish is something more modest and more durable, that the density and quality of spindle activity during NREM sleep are not fixed, not simply a passive readout of some underlying state. They are influenced by the brain's own prior activity during the waking hours, and they can be shifted by carefully designed external intervention. The spindle is not a given. It is a process that the sleeping brain can do more or less of, better or worse, under different conditions. Targeted memory reactivation takes this logic a step further. The idea begins with an observation so simple it is almost surprising that it works. If a memory was encoded during waking in the presence of a specific sensory cue, a sound, a smell, then presenting that cue during NREM sleep, quietly enough not to wake the sleeper, can preferentially reactivate the memory trace associated with it. The cue reaches the sleeping brain not as a message it can consciously decode, but as a familiar signal that briefly biases the pattern of neural activity toward the encoding era state associated with it. The foundational experiment, published in 2009, used a spatial memory task in which participants learned the screen locations of various objects, each paired with a characteristic sound, a cat appearing at a particular location, accompanied by a meowing sound, a kettle somewhere else accompanied by a boiling sound. After learning, participants slept in the laboratory. During confirmed NREM sleep, the computer softly replayed half of the paired sounds. The other half were withheld. In the morning, the participants were tested on their memory for all object locations. The locations associated with the replayed sounds were remembered significantly better than the locations whose sounds had not been played during sleep. The sleeping brain had used the cue. It had done something different with the memory trace associated with that sound, and the effect was not present when cues were played during waking rest or during REM sleep, only during NREM. Subsequent work refined the timing. Cues delivered during detected sleep spindles or precisely during detected slow oscillation upstates produced larger memory benefits than cues delivered at random intervals during NREM sleep. The oscillatory structure of NREM was not merely permissive, it was load-bearing. The same cue, carrying the same familiar signal, produced different outcomes depending on whether it arrived during an active spindle or during the quiet between spindles. During an active spindle, the cortical circuits were in their optimal state for receiving and processing hippocampal replay. During the quiet intervals, the same signal landed without the same purchase. This is a finding with implications for what a spindle is, functionally, in the sleeping brain. Each spindle is not just a burst of rhythmic activity, it is a brief opening, a window during which the thalamocortical circuit is organized, synchronized, and ready to receive the compressed replay that the hippocampus is simultaneously generating. Outside the spindle window, that readiness is reduced. Outside the slow oscillation upstate, the window is mostly closed. The three-frequency hierarchy, slow oscillation, spindle, ripple, is not three separate processes occurring near each other in time. It is one integrated system, tightly coupled, each element depending on the others to do its work. The precision of that coupling is something worth sitting with for a moment. The slow oscillation completes less than 1 cycle per second, a great rolling rhythm whose half cycle, the upstate, lasts only a few hundred milliseconds before the cortex falls quiet again. The spindle completes 12 to 15 cycles per second, faster, nested inside that brief upstate, its entire waxing and waning shape compressed into a second or less. And inside the spindle, at the trough of the oscillation, when the thalamus briefly re-inhibits the cortex, hippocampal ripples arrive at 80 to 100 cycles per second, compressed bursts so brief that each one lasts less than a tenth of a second, carrying within it an entire replay sequence from a prior waking experience. 3 rhythms, 3 different time scales, all 3 aligned during each NREM cycle, thousands of times per night in the dark interior of the sleeping brain. The process has no witness. The sleeping person feels none of it. The experiences being replayed in those ripples, a face encountered during the afternoon, a route walked, words learned by effort at a desk, pass through the system without producing any awareness, without producing dreams in any ordinary sense, without disturbing the slow, quiet breathing of the sleeping body. All of it happens below the surface, conducted in the rhythmic electrical language of neurons, rather than in anything that has a name in waking experience. What it leaves behind is less dramatic than the process itself. It leaves slightly stronger synaptic connections between neurons that were active together during the waking event. Not dramatically stronger, not transformed, just a fraction more likely to fire together the next time the right input arrives. Multiplied across thousands of replay events across a night of sleep, across dozens of synapses per event, across the hippocampal neocortical network that holds the distributed traces of a day's experience. That slight strengthening accumulates into the difference between forgetting and remembering. The memory is not written during sleep. It was written during waking. Sleep, and the spindles within it, consolidates what was written, makes it more stable, more resistant to interference, more likely to be accessible tomorrow. The selectivity of that process matters as much as the process itself. Not all of today's experience is preserved with equal fidelity. The brain is not a recording device. It does not store everything that passed through its circuits today. During waking, something marks certain experiences as worth keeping, a signal that is not yet fully understood, but that likely involves the same dopaminergic and cholinergic modulation that accompanies attention, novelty, and emotional salience during conscious experience. By the time sleep arrives, certain synapses have been tagged, transiently, at the molecular level. Spindle-driven calcium influx during sleep preferentially strengthens those tagged synapses. The untagged ones, the unremarkable, inattentive moments of the day, do not benefit in the same way. The spindle is not indiscriminate. It consolidates what was marked for keeping. This is the picture that has emerged from decades of work, refined through correlational studies, experimental interventions, pharmacological manipulations, and the slow accumulation of converging evidence from laboratories that approach the question from different angles. It is not a complete picture. There are aspects of spindle function that remain unclear. The precise molecular mechanisms by which calcium influx during a spindle interacts with synaptic tagging proteins, the question of how the brain decides which spindles to generate over which cortical regions, the degree to which spindle enhancement in clinical populations might restore consolidation deficits without unwanted effects. The picture has open edges. That is the normal condition of any science that is still alive. What is settled is substantial. Sleep spindles are generated in the thalamic reticular nucleus through a well-understood circuit mechanism. They occur with highest density during stage 2 NREM sleep. They couple in a precise temporal hierarchy with slow oscillations and hippocampal sharp wave ripples. That coupling provides the synaptic timing conditions required for hippocampal neocortical memory consolidation. People who generate more of them remember more. boosting them, whether through closed-loop stimulation or pharmacologically, improves memory retention. The effect is preferential for hippocampus-dependent declarative memory, though procedural memory benefits have been documented under certain conditions. Spindle characteristics are partially heritable, decline with age, and vary stably across individuals in ways that predict cognitive performance independent of waking factors. In the sleep laboratory, the night continues. The researcher at the monitor has been watching for several hours. The sleeper has cycled through deep, slow-wave sleep twice, passed through 2 light NREM episodes, and entered a second long REM period that is now drawing to a close. The body will descend again in the next few minutes into stage 2 NREM. The sigma band will quicken, the spindles will return. They will cluster across the next 30 or 40 minutes in brief high-density bursts, separated by intervals of relative quiet, their exact timing driven by the oscillatory dialogue between the thalamic reticular nucleus and the thalamocortical relay neurons and the cortex, a dialogue that has been running without interruption since the sleeper's eyes closed hours ago. The researcher has seen this pattern hundreds of times. The trace on the monitor is familiar to her in the way that a coastline is familiar to someone who has watched the tide come and go for years. She knows what the spindle looks like before it fully appears. The slight tightening of the sigma band, a half second before the amplitude rises, the waxing shape unfolding at its characteristic pace, the crest arriving just as the thalamocortical circuit reaches the peak of its rhythmic excitation, and then the gradual return to baseline as the inhibitory loop re-engages and the burst subsides. She is not watching with urgency. There is nothing to intervene in, nothing to prevent. The sleeping brain is doing exactly what it is built to do. What she is looking for, in the long, attentive way that scientific work requires, is the detail that has not been fully accounted for yet. The spindle that does not follow the expected pattern, the coupling event that falls slightly outside the predicted timing window, the parameter that shifts in a way the current model does not predict. The next refinement of the picture, when it comes, will likely begin with a moment like this one. A quiet night, a long record, and a small discrepancy that someone trained to notice it did not look past. Outside the laboratory building, the night is at its stillest point. The city is as quiet as it gets. The hours between 2 and 4 in the morning carry a particular quality of silence, not the silence of early evening when the day has only recently ended, but the deeper silence of the night's interior, when even the light traffic has thinned to almost nothing, and the air holds a quality of suspension, as if the ordinary momentum of the day has not yet begun to gather again. The sky overhead, where it is visible between buildings, holds its cold and ancient depth, unmarked by anything happening in the room below it. Inside, the monitor continues its long green trace. The sleeper breathes in the dark. The slow oscillations move through the record like long, low rollers crossing a still sea, and within them, at the rising phase of each slow wave, the spindles appear and fade, each one a half-second cluster of sigma frequency oscillations, waxing and waning in the characteristic shape that makes them unmistakable among all the other rhythms of the sleeping brain. Each one a brief organized burst in the thalamus, fanning upward through thalamocortical fibers into the cortex, carrying with it the conditions under which a fragment of today's experience can be made a little more durable against the attrition of time. There is no drama in this. There is no moment of completion, no threshold crossed. The consolidation of memory during sleep does not announce itself. It accumulates, quietly and continuously, across each NREM cycle in the same way that small deposits of sediment accumulate on the floor of a slow-moving river, not in any single event, but in the sustained, unhurried work of many small additions over a long time. By morning, the record will show 4 to 6 complete sleep cycles, each with its slow wave valleys and its lighter NREM plateaus and its REM peaks. The spindle count across the night will run to several hundred events. The word pairs learned the previous evening will be slightly more accessible than they would have been without the night's sleep. The spatial memories will be slightly more stable, the procedural sequences slightly more automatic. None of these improvements will be felt directly. The sleeper will not wake, knowing that consolidation occurred.